Okay, so this is, I think this is the wrong one. Hold on a second. Sorry about that. I'm going to stop the share for a second. I'm going to have to go back. It won't let me minimize. I have to stop the recording. Okay, that's a big question now. How do I stop recording it? Okay, unfortunately, there's going to be a gap there. Will I find the right video? I mean, the right lecture. Let me double check that. Let me just double check this. I think we're doing a different lecture. I just want to make sure I'm going with what's going to be on the test. Actually, we do want to do chapter 5. Sorry about that. Chapter 7 will be in the next lecture. So I'm sorry I got thrown off there. We do want to do chapter 5. Let me go back. Okay, sorry about that. I was thinking that this was chapter 7, which will be in our next section, and my description was correct. We are doing chapter 5. So can everybody see that?
Yes.
Yes, Professor.
Okay. All right, thanks for your patience. So this is, we're going to start talking now just a little bit about the history of eukaryotes in terms of evolution. And we'll talk about eukaryotes, and then we'll talk about some, as I said before, protozoan and other eukaryotic parasites that cause disease. So the major groups, the bacteria, the archaea, and eukaryotics, we're going to talk a little bit about their evolution. And let me omit these people who just joined. As you know, the bacteria, archaea, are single cell or unicellular, and prokaryotes are not multicellular. So eukaryotes can be single cell or multicellular. We believe the first eukaryotic cells appeared 2 to 3 billion years ago. Bacteria and eukaryotes evolved from a common precursor called the last common ancestor. This early ancestor was neither pro or eukaryote. It gave rise to bacteria, archaea, and eukaryotics separately. Organelles is something that developed later out of primitive cells that became trapped in eukaryotic cells.
Professor, can you go back, please?
Sure.
Thank you. Thank you. I got it. You can see.
Okay, no problem. So this primitive cell probably started having its genetic material was RNA. DNA is more complicated. And it gave rise to archaea and bacteria. At some point, there was an endosymbiotic relationship, which primitive cells, the bacteria, either photosynthetic bacteria or another type of bacteria, became actually an organelle inside a larger cell, which gave rise to a eukaryote. Remember, bacteria do not have these organelles. Our mitochondria and the chloroplasts in plants have remnants of that. They actually have their own genetic material. The mitochondria and the chloroplasts are actually more similar to bacteria. They replicate independently of the rest of the cell. First, primitive eukaryotes were single cell and independent. They became specialized to perform a particular function in a colony. The colony multicellular organisms evolved when the individuals lost the ability to survive apart from the colony. This was probably an early precursor of differentiation of labor in a system, leading to tissues and eventually organs. So the protozoans, which are eukaryotes, they are almost always single cell or unicell, means same thing. Fungi and algae that are eukaryotes, some of them are single cell and some are multicellular. Helminths that we're going to talk about this evening, their egg is single cell or sometimes in the larval form, but the adults are always multicellular. Eukaryotes, true or false? Eukaryotes and prokaryotes evolved independently of each other.
Is that true?
No, that's false.
Oh.
There was a common ancestor for both, and prokaryotes probably evolved from an endosymbiotic relationship. That means the symbiosis was inside the cell. So we still have that. Our mitochondria has a circular DNA. It has its own ribosomes, its own genetic material to a certain extent. In fact, in terms of molecular evolution, a lot of times the ancestors are traced down through the genetic material in the mitochondria. Blank evolved from primitive cells that became trapped in larger cells.
Organelle?
The eukaryotes evolved from primitive cells that became trapped in larger cells. The difference between tissues and organs. So our organs are specialized structures that are made up of different tissues. which in turn are made up of different cells. We're going to talk, list 3 types of eukaryotes that we've already mentioned, studied by microbiologists. We just mentioned some of those.
Yeah, algae, fungi, and
Fungi, and
algae.
what was another one?
Algae, and then protozoa. I can say that word.
Yeah,
Protozoa.
no, protozoans. You're correct. That's right. So we're going to talk about structures of bacteria, archaea, and eukaryotes that are similar. We're going to talk about the importance of a glycocalyx in eukaryotes. In bacteria, we call it a capsule. List which eukaryotes might have a cell wall. Some of them do have cell walls. It's made of a different material than what bacteria's cell walls are made up of. Talk about differences and similarities between eukaryote and bacterial cytoplasmic membranes. So if you remember the basic cell membranes, a phospholipid bilayer. So in eukaryotic cells, they have a cytoplasmic membrane. They have nucleus, they have mitochondria, they have an endoplasmic reticulum, Golgi apparatus, they have vacuoles. The cell structure itself has a cytoskeleton made of proteins, and they can have a covering called a glycocalyx. Glyco for sugar, calyx, I believe, means just coat. Some eukaryotic cells have a cell wall. Some of them have appendages for movement. They have flagella, but it's made of it's made a little differently than the bacteria flagella. And some eukaryotic cells are chloroplast. So basic, generalized eukaryotic cell. It has all these membranes inside of it, mitochondria. It could have chloroplast for photosynthesis. It can have a Golgi, lost my cursor there, Golgi apparatus. It can have these vesicles called lysosomes, which also our phagocytic cells have. And actually in the process of phagocytosis of a bacteria, these play an important role. They have filaments and proteins that are the cytoskeleton to help give it structure. They have a nucleus, which is surrounded by membrane. Coming out of that is the endoplasmic reticulum. And you can see these little dots in here represent ribosomes. It's the site of protein synthesis. Bacterial cell, remember they have a single circular chromosome that's localized to a certain region, but is not bound by a membrane. Bacteria can also have the flagella and these pili for attachment. Eukaryote versus bacterial flagella. The eukaryotic flagella is 10 times thicker, more structurally complex. It's covered by an extension of the cell membrane. It has long sheath cylindrical structures containing regularly spaced hollow microtubes. It has a what's called a 9-2 arrangement. So you can see that here, you see 2, 1, 2, 1, 2. You count all these up, it should be 9. And seems to be true. Cilia is a similar structure to flagella. It is short and more numerous, and up to several thousand of them are seen in cells. They are found only in a single group of protozoan in certain animal cells. They function in feeding and filtering on some cells. We also have cilia in our respiratory tract that help beat and move the mucus upwards. The glycocalyx, a boundary that comes in direct contact with the environment. It's also called the extracellular matrix. Actually, multicellular organisms like us have an extracellular matrix with various molecules in it. The glycocalyx in the prokaryotes, in the eukaryotes, composed of polysaccharides. It appears as a network of fibers. It has a slime layer, and it's similar to a capsule. The cell wall is seen also in fungi and algae. It is cell wall in plants is made of cellulose. The cell wall in bacteria is made of peptoglycans. And fungi, it's made of chitin, which is also the same thing that makes up the extracellular, the exoskeleton of arthropods, like insects and lobsters, for example. It is rigid and provides structural support in fungi and algae, different chemical composition. It is either chitin or cellulose in fungi. But the hallmark of, even though these are all different, they still retain some sort of phospholipid bilayer in the cells. Cytoplasmic membrane, as I said, it's typical bilayer phospholipids, has protein molecules in it. It contains sterols to stabilize the eukaryotic membrane. It is a selectively permeable barrier, sophisticated mechanisms for transporting nutrients in and waste products out. What are three ways in which eukaryotic flagella are different than prokaryotic flagella? It's bigger and thicker. What else? It has those microtubules in twos, and then there's 9 of those, the 9-2 structure. Functions of a glycocalyx. It is a protective barrier to prevent drying out of the cell. What were some of the other functions? Let's go back to the glycocalyx. Actually doesn't tell you the functions, but its appearance, it's made of fibers, slime layer, capsule. Oh, the cell walls. Eukaryotic cell walls can be made of chitin or cellulose. In bacteria, they're made of peptoglycan. We're going to talk about some eukaryotic
Do they
internal.
contain cellulose?
Pardon?
I say, do they contain cellulose?
The plants, the
Mm-hmm.
cell wall's made of cellulose. In fungi, it's made of chitin. Was that your question?
Mm-hmm. Yeah, thank you.
Okay. So we're going to talk about internal structures in eukaryotes, the nucleus, the endoplasmic reticulum, Golgi apparatus, and vesicles that are used to transport things. Talk about the mitochondria. We'll talk about the ribosomes. So in our metabolism section, I believe in our next section, we'll be doing electron metabolism. We'll talk a little bit more about the mitochondria. In our molecular biology, genetics, we're going to talk about ribosomes and protein synthesis. Bacteria and eukaryotes both have ribosomes. They're a little simpler in structure in bacteria. We'll talk about the cytoskeleton, which is proteins inside the cell to help give it some structure. And we've already mentioned how endosymbiosis led to the development of eukaryotic cells. The nucleus is a compact sphere in eukaryotes. It's the most prominent organelle. It's separated from the cytoplasm by a membrane called the nuclear envelope. There's a nucleolus region that stains intently for RNA. That's where transcription of RNAs occurring, and also ribosomal RNA, which are necessary to carry out protein synthesis. Bacteria divide by binary fission. Eukaryotes can divide by either mitosis or meiosis. Bacteria do not undergo meiosis. In mitosis, the genetic material has to be duplicated before the cell separates to make a new cell. In meiosis, you get the production of the sex cells, but the genetic material is no longer what we call diploid. It'll be a single chromosome until the egg cell is formed. Microscopic, the endoplasmic material, reticulum, is a series of tunnels for transport and storage inside the cell. We have what we call the rough endoplasmic reticulum, where ribosomes are attached to the surface. This is where protein synthesis occurs. The proteins are then held for packaging and transport. Smooth endoplasmic reticulum does not have ribosomes, and it is a site for the synthesis and storage of non-protein molecules. So the rough endoplasmic reticulum is showing you both a diagram here and an actual electron micrograph of the nucleus here and the endoplasmic reticulum here, covered with ribosomes. Ribosomes are made up of two units, both in eukaryotes and prokaryotes, that comes together. Then what's called the message RNA binds, called mRNA, binds to the ribosome, and that starts protein synthesis. The proteins then go out to the endoplasmic reticulum. The Golgi apparatus, which is a continuation of these membranes that come off of the nucleus, is a site of protein modification and transport. Consists of a stack of flattened disc-shaped sacs closely connected to the endoplasmic reticulum. It is where it is close to the Golgi apparatus, the endoplasmic reticulum forms transitional vesicles that are picked up. So in other words, proteins. Sorry, dog keeps Proteins are synthesized on the endoplasmic reticulum. They again get transported to the Golgi apparatus through these vesicles. There in the Golgi apparatus, polysaccharides and lipids can be added to the proteins, and more vesicles are formed, conveyed to lysosome, where they are transported as secretory vesicles. Details of the Golgi apparatus. So you have the endoplasmic reticulum where the protein synthesis occurs. They then go through these vesicles to the Golgi apparatus where they can be modified, and then they go to these condensing vesicles that come off of the Golgi apparatus. The nucleus, the endoplasmic reticulum, Golgi apparatus, it's like, think of it like an assembly line. The genetic information flows from the nucleus, from the genetic code on the DNA. Proteins are synthesized on the ribosomes on the endoplasmic reticulum. Then they are transported to the Golgi apparatus, where they are modified and packaged into vesicles. So this shows you the genetic material. The synthesis of RNA is occurring in the nucleolus. The proteins then go to the, I should say actually the message RNA goes to the endoplasmic reticulum, and then the proteins are synthesized, packaged into vesicles, transitional vesicles that go to the Golgi apparatus. And then from these vesicles, they can be secreted by exocytosis into outside of the cell. Some other specialized cellular vesicles. Lysosomes originate from the Golgi apparatus and have a variety of enzymes. We'll see those that when we talk about phagocytosis in the immunology section. These are involved in intracellular digestion of food and protection against invading microorganisms. Vacuoles are membrane-bound sacs that contain fluids or solid particles to be digested, excreted, or stored. Mitochondria, energy is generated in the mitochondria. The bulk of the energy of the cell is generated in the mitochondria. It has two basic structures, a cristae that is made up of folds of an inner membrane. This membrane holds enzymes and electron carriers for what we call aerobic respiration. Aerobic respiration leads to the generation of energy that the cell can utilize. The matrix of the mitochondria has ribosomes, DNA, and enzymes and other compounds used in metabolism. Unique characteristics of the mitochondria. As I said, they can divide independently of the cell. They actually have circular DNA like a bacteria. They have bacterial-sized ribosomes. All this provides evidence that mitochondria were cells that they were an endosymbiont engulfed by other cells and became cellular organelles. Chloroplasts found in photosynthetic cells. They convert light energy into chemical energy. They're the primary producers of all organic nutrients. So the cells, photosynthetic cells, convert the light energy into glucose and give off oxygen gas to the environment. We use the glucose in complex molecules to generate energy, and we give off CO2, which the plants use the CO2 and light energy to make the compounds. Protein ribosomes is where the protein synthesis occurs. They are distributed throughout the cell. They're in the cytoplasm, in the cytoskeleton of the cell. They attach what's called the rough endoplasmic reticulum. They're also found in mitochondria and chloroplasts. When you have a chain of ribosomes, it's called a polyribosome. Eukaryotic ribosomes are similar to bacterial ribosomes in structure. They have both a large and a small subunit. The ribosomes are made up of both RNA and protein. A full-size ribosome in eukaryotes referred to as 80S. Did somebody ask a question? And it's made up of two subunits, a 60S and 40S. These don't refer to the weight, but a sedimentation coefficient. So in eukaryotes, it's 80S, and a prokaryote, it's 70S. Major elements of each type organism. So bacteria, archaea, and eukaryotes, they all have some sort of nucleic acid as the genetic material. Bacteria and archaea and eukaryotes all have chromosomes. Viruses do not have chromosomes. Viruses do not have a nucleus, and they do not have a nuclear envelope. And neither do bacteria. Bacteria and viruses do not undergo mitosis. Eukaryotes do. Bacteria, some can make a sort of like they can exchange genetic material, but only eukaryotes undergo true meiosis. Bacteria divide by primary fission. Viruses don't. Eukaryotic cells divide by mitosis. I don't know why they're saying here binary fission. Biosynthesis can occur, but not in viruses. Viruses do not metabolize. They don't have a Golgi. They don't have endoplasmic reticulum. They don't have ribosomes. Mitochondria are only found in eukaryotes. Chloroplasts are only found in photosynthetic eukaryotic cells. Flagella can be found in bacteria, and they can be found in some eukaryotic cells. Cilia is something found in eukaryotic cells. Again, viruses don't have that. Viruses don't have a proper cell membrane. They can take the membrane of a host cell and put their own proteins in it, but they do not synthesize their own cell membrane. They do not have a cell wall. They do not have any capsules. The size differences, you can see that bacteria are smaller in a eukaryotic cell. In fact, some bacteria are about the same size as the mitochondria. Viruses are very small. Eukaryotic chromosomes, true or false, are visible during all portions of the cell cycle. So when the cell divides, there are parts of that cell cycle that you can see the chromosomes.
No, that's false.
Pardon?
I said false.
I'm sorry, I couldn't quite hear you.
I said false.
False. Okay.
Yeah.
So the endoplasmic reticulum, I'm sorry, I should say the blank endoplasmic reticulum is responsible for packaging and transport of proteins. What was that term? What was the difference between the rough and smooth endoplasmic reticulum? So the rough endoplasmic reticulum has ribosomes on it. It's an area of active protein synthesis. Blank vesicles contain digestive enzymes. You remember what vesicle when we were talking about that? You can go back and look at that.
Is it lysosome?
It is the, oh, how far back is that? I must have skipped over it, sorry. But yeah,
Is
the
it the
lysosome has digestive enzymes in it. You were correct. Okay, we're going to somebody needs to mute themselves, please.
Oh, I'm sorry.
Okay, thank you. Okay, we're going to talk about fungi, the basic structure. Remember, fungi can be single cell or multicellular. We're going to talk about a heterotroph, a saprobe, and a parasite. We'll talk about the relationship between fungal hyphae and the production of the mycelium, and two ways in which fungal spores can arise. We'll talk about two detrimental and two beneficial activities of fungi from our viewpoint as humans. There are approximately 3 to 4 million species of fungi. The two major groups, the macroscopic fungi, you can clearly see those, mushrooms, puffballs, gill fungi. Microscopic fungi consist of molds and yeast. A yeast shell cell is round to oval shape. It uses asexual reproduction. You can have these long hyphae that are made up of cells. Seen this in filamentous fungi and molds. So these hyphae are branches, but they're connected cells. Yeast can make like a pseudo hyphae. It's similar, where these separate or at areas called septa. Some fungi can be dimorphic. They can take either form, whether it's that or in the, I think what they're referring to, or in the hyphae. So yeast are a eukaryotic single-cell organism. They have all the internal structures of a eukaryotic cell: mitochondria, a nucleus, endoplasmic reticulum, Golgi apparatus. I believe I already mentioned the mitochondria. One way they can reproduce is they just simply bud off a new cell. And so you see these little buds that appear, and they eventually bud off. They can be connected in a chain called a pseudo hyphae. So fungal nutrition, there are ones that are heterotrophic. They require nutrients from a variety of substrates. There's ones that are saprobes. What they do is they secrete enzymes into the environment, and their substrate is dead plants and animals that they digest the material outside of the cell and transport it in. There are ones that are parasites, and they have to live on a living plant or animal. So an example of that is athlete's foot, which is a fungus. Most microscopic fungi grow in loose associations or colonies. They can have a mycelium, which is a woven intertwining mass of those hyphae filaments that makes up the body or colony of a mold. The septa is where walls between dividing hyphae form. And these spores that they make are actually reproductive bodies to make new fungi. So this is the bread mold. This would be basically a mycelium structure of the mold. They can have these hyphae, if you were to enlarge this, or they can have structures that will then release spores that will create more of the fungi. Fungi have many complex and successful reproductive strategies. They can simple outward growth of the hyphae. The mycelium can separate and generate new colonies. Their primary, reproductive mode is to make spores. Fungal spores shouldn't be confused with the more resistant non-reproductive bacterial endospores. So these are not like endospores, though they can be a resistant structure, but they are not as resistant as a bacterial endospore. Sporangiospores are the is the term for the structure in a fungi that makes the spores. So it can be either in a membrane sac or not enclosed by a spore-bearing sac. So I'm sorry, the membrane, the structure that makes the membrane enclosed spores is called the sporangiospore. And the condiospores are the ones that are not surrounded that structure. Sexual spore formation is important when fungi or different genetic makeup combines for their genetic material. Slight variation in form and function are potentially advantageous to evolve and adapt in the survival of species. Place again. Please be aware of muting yourselves. So if you unmute to ask a question, that's great. But once you're done, try to remember to mute yourself. So fungal identification and cultivation. Identification of medical specimens is important. Isolation, like we do with bacteria on special media. A little easier to observe either microscopically or macroscopically. Asexual spore-forming structures and spores can help identify the genus and species of fungi, along with the hyphae type, colony texture, pigmentation, physiological characteristics, and their genetic makeup. You can see those two spore-bearing structures, the sporangium that's surrounded by membrane, and the condius, I think it was called spore, which isn't surrounded by a membrane. Most medical conditions caused by fungi, most fungal infections occur through accidental contact with soil, water, and dust. There are pathogenic fungi, community-acquired infections from environmental pathogens, hospital-associated infections caused by pathogens in the clinical setting, and opportunistic pathogens. These are caused by pathogens that have already infected a weak individual. Other medical conditions caused by fungi, people can have allergies to them. They can even have neurological conditions due to toxins made by the fungi. Important role that bacteria play in the environment. They are important in decomposing of organic material and returning the nutrients to the soil. Fungi also make stable association with plant roots that helps them increase water and nutrient absorption. Fungi can make antibiotics. They can make alcohol, organic acids, and vitamins. They're used in food flavoring, alcohol, in beer, wine, in the gas, in bread is all due to fungi. Didn't mean to go back. So a blank is a type of fungus that derives nutrients from dead or decaying material. You remember what that is? It starts with an S. Saprobe. True or false? Fungal spores have a tough outer coating that allows them to survive in extreme conditions.
False.
Yeah, it's not like an endospore. It can be a resistant structure, but not like an endospore. Wouldn't survive extreme conditions. Blank or asexual spores contained in a sac-like head. What was those called? It's the sporangio.
Sporangium.
The sporangio. I'm probably not going to.
Sporangius.
That's right. And true or
Sporangium.
false? Sporangium. Yes.
What is the sparing?
And lastly, true or false? Pathogenic fungi require a host to complete their life cycle.
True.
Say it's true. They didn't actually, I don't remember them mentioning that specifically, did they? Sporangiospores, candiaspores. Okay, this next section are going to be the protists, but let me stop for a section, stop here at this section to double check the roll. Is Felicia Adams here? Is Chantel Allen here?
Yes, I'm here.
Okay. Is Tanisha Allen here?
Here.
Okay. Holly Cogburn, are you here?
Yes.
Okay. Donasco.
Here.
And you're here. Sheena Howard. Malone McDaniel.
Yes, sir.
Okay. Natalie Smith. Kiesha Talley.
Natalie's here. She just stepped away for a moment. She's here though.
Natalie Smith is here. Okay, sort of need to hear it from her. Was Kiesha Talley here? Is Wallace here? Let's see. Okay, so if I didn't call your name, I've already marked you as present. I'll check
Okay.
one more time at the end of the lecture.
I just want to.
Yes, go
I just
ahead.
want to make sure, Professor, you do have me marked absent. I mean, marked present.
I'm sorry, who's this? Who was just asking me?
I'm here, Professor.
I'm sorry, who's this?
Brenda Copy.
Brenda? What's your last name? Oh, Brenda Coffey. Yeah, I've already marked you as
Coffey.
present. Yeah, sorry.
You already marked me, Professor Deidre Francis.
Yes. I've already marked you as being present.
I'm here too. Asiatu.
And who's that?
Asiatu.
Yes, I've
Did
already
you get
marked.
Natalie Smith?
Yes, no. I mark you present now.
What about Charlotte, Professor?
Charlotte, did you say?
Yes.
Or Chantel? You said Charlotte?
Yeah, Charlotte Zokonyan. Last name Zokonyan.
Oh, I'm sorry. I always, yes, you're the last one because it starts
Yeah.
with D. I've already marked you as present. I'll double check again. So I think this is roughly the halfway point. We're going to take a 5-minute break, roughly come back around 8:06. So 5 minutes, we'll come back.
Am I the only one or just not talking?
We have a break for
It's a
5.
break.
Come back after
Oh,
5.
okay. Thank you.
You're on mute, Professor. Professor, you're on mute.
I'm sorry, I thought I unmuted myself. So we're going to talk in this section about the protozoans. Their characteristics that put them into a single group. 3 ways they can move. Talk about the cis stage, which is a resistant structure in protozoans, and talk about some diseases caused by protozoa. Algae and protozoa have traditionally been combined into the kingdom of Protista, and then that is divided into 2 subkingdoms, one for algae and one for protozoans. A protist is any eukaryotic single cell or colony forming organism that lacks true tissues. Algae are photosynthetic organisms like seaweed and kelp. They are most recognizable. Algae exhibit all the eukaryotic organelles. They have chloroplasts like
The top one I'm putting eating the core. I don't know about the
Mute your mic, mute your mic.
okay. There's other pigments that can be seen in algae that are yellow, red, and brown in coloration. Algae are widespread inhabitants of fresh and marine water. Plankton are floating communities of microscopic organisms. They play an essential role in aquatic food web and produce most of the Earth's oxygen. The primary medical threat from algae is through ingestion of toxins during a red tide. Now, for protozoans, there are 65,000 species. Most members are harmless inhabitants of the water and soil. A few species are parasites responsible for hundreds of millions of infections in humans every year. Single cell containing all the major eukaryotic organelles, except for chloroplasts, are seen in the protozoans. They can have some organelles that are highly specialized. They look like mouths. They have digestive systems, reproductive tracts, and even appendages for movement. They are heterotrophic and require their food in complex organic form. There's free living species that scavenge dead animals or animal debris. They graze on living cells of bacteria and fungi, algae. There are parasitic species which live on fluids of a host, such as the plasma and digestive juices, and they can actively feed on tissues. The ways that they can,
I have a question.
yes.
Did you mean that they so compared to the other one, they don't feed themselves?
What do you mean by feed
You
themselves?
said they require food in complex organic free living species. Can you explain that?
They require, so a heterotroph, and we're heterotrophs, we require food that's complex molecules.
Oh, okay.
Does that make sense? So
Mhm.
we carbohydrates, proteins, lipids.
Oh, okay. Got it. Thank you.
Okay. So ways that they can move, they can make these pseudopods, which are false feet that amoebas use. They can also serve as feeding structures. They can have flagella, though we mentioned before, it's a different structure than the one bacteria have. They have cilia over the entire surface of the cell. So these are some examples. You can see the flagella in this protozoan. You can see the pseudopods being formed by this amoeba that enables them to move. They can have the cilia on their surface, and they can have these food vacuoles on the cell. So in their life cycle, they have a feeding and a dormant stage. During the feeding stage, they're called a trophozoite. That's when they're mobile and they're feeding. They require food and moisture to remain active. If they come to an unfavorable environment, they can form a cyst. The cyst is a dormant resting stage formed in an unfavorable growth environment. And these cysts can also be an important way that disease can be spread from protozoans. So they can have, starting here, an active feeding stage called the trophozoite. Coming into an unfavorable environment, they will start making a wall and make a mature cyst. It's a dormant structure. And then when there's nutrients available and moisture, like the endospore and bacteria, they can, this can break down and they can release a new trophozoite. The life cycle of protozoans can be simple to rather complex. Some protozoans exist only in the trophozoite stage. Many alternate between this stage and a cyst stage, depending on the conditions of the habitat. The life cycle dictates their mode of transmission. Protozoan reproduction can be simple, asexual means, usually by mitosis. Several parasitic species reproduce by multiple fission. Sexual reproduction can also occur. They can also carry out conjugation, which is similar to bacteria, where they can exchange genetic information between two cells. They can be classified. This classification of selected protozoan, they can use flagella to move. They can be classified as flagella alone or flagella and amoeba to motion. They can have a single nucleus. They can have cyst and are also free living. Some use amoeba motion. Some use pseudopods. They can have asexual reproduction by fission. Most have a single nucleus, but some have two nucleuses. They usually make a cyst, in other words, and are free living and not infectious. So those that use cilia to move have cilia and tufts for feeding and attachment. Most develop cysts, most have a macronuclei and a micronucleus. They divide by something called transverse fission. Most have a definitive mouth or feeding organelle. They are free living and harmless. Those with no motility are the sporozoa. Motility is absent in most representatives. They have complex life cycles with well-developed asexual or sexual stages. This entire group is parasitic. Most protozoans can be identified at the level of genus because of the unique appearance, shape and size of the cell, type and number of their locomotive structures, presence of special organelles or cyst, and the number of nuclei they have. They can infect blood, septums, cerebral spinal fluid. They can be found in fecal matter or vaginal specimens. They can be smeared on a slide with or without staining. So parasitology is the study of these protozoans along with parasitic worms, the helminths. Parasites, the term most often used to characterize these pathogenic protozoan in worms. So some examples: Entamoeba histolytica. It's an intestinal parasite, contaminate humans, water, and food. Acantha amoeba can cause brain infection, found free living in water. Some ciliated protozoan, the Bolantidium coli, is also an intestinal parasite, infects pigs and cattle. Some other ones, I won't go over all of these, but Giardia is a common one. Our dog got it from just eating stagnant water when we went on walks. So it's found a lot in these small puddles of water. Trypanosomes, they can be found in animals or vectors, can cause sleeping sickness, Chagas disease. Let's see down here, Plasmodium, an important one, causes malaria, cardiovascular and other symptoms. It can be transmitted by mosquitoes. Toxoplasmus, it's a flu-like illness, toxoplasmosis. Some pathogenic flagellans have flagella. The trypanosoma, it causes sleeping sickness in Africa. And the one that causes Chagas disease is endemic to South and Central America. They have long crescent-shaped cells with a single flagellum. They're both found in the blood during infection, and they're transmitted by blood-sucking vectors, in other words, insects. So, true or false? Algae don't cause any type of human disease. Is that true?
True.
That isn't true, though only a small group. It's usually not a problem, but there are some algae that can cause human disease. What are the 3 ways that protozoans can move? They have those pseudopods, kind of like feet-like structures. They can have flagellum. And what was the 3rd way?
Is it like a boat?
Cilia.
The cilia. True or false? All protozoans have a trophozoite form, but not all produce cyst. Is that true or false?
False.
No, that's true. They have a mobile feeding phase, but not all of them produce cyst. The term blank is most often used to describe protozoan and helminth pathogens. Here,
Parasite.
correct.
What did you say?
That's correct.
Parasite.
Number
Parasite.
4 is parasite. We're going to talk now about the parasitic worms, 2 major groups, provide some examples and the stages of the typical life cycle. So helminths are consistent tapeworms, flukes, roundworms. These are usually large enough to be seen with the naked eye. They can be 1 millimeter to 25 meters in length. They usually have a structure at the end for attachment and deriving nutrients from the host. They are multicellular. They even have organs and organ systems. They have a reproductive tract in the most developed ones. They have primitive digestive, excretory, nervous, and muscle systems. They have thick cuticles for protection. They have mouth glands for breaking down the host tissue. Their life cycle consists of a fertilized egg, which then becomes a larval, which then becomes an adult. The majority of helminths derive nutrients and reproduce sexually inside the host body. Of the different types, nematodes, the sexes have different morphologies. The trematodes, the sexes are separate or hermaphroditic, which means they can go from male to female. Or actually, the next sentence says that better. They can have male and female sex organs in the same worm. Cestatodes are generally hermaphroditic. Their general life cycle is transmission of an egg or larva to the body of another host. They have sometimes an intermediate secondary host in which the larval development occurs. And then they have a definitive final host in which the adult form and mating occurs. They can be found in food, soil, water, infected animals, routes of infection, oral intake or penetration of unbroken skin. Humans are the definitive host for many species and the sole biological reservoir for about half of the diseases. Animals and insect vectors can also be reservoirs for these parasites. So roundworms are the nematodes. Some common ones you might have heard of, pinworm. They require a human host. They can be spread by fecal pollution of soil with eggs and ingestion of these eggs. It gets into the water. The trichinella worm found in pigs and wild mammals. You can get it from consumption of meat containing the larvae. The tissue nematodes cause river blindness carried by black flies, and you can get it from a fly bite. A guinea worm, ingestion of water. In this one called Cycloops is an aquatic invertebrate that carries the guinea worm larva. some flatworms, blood fluke found in humans and snails, ingestion of fresh water containing the larval stage, pork tapeworm, consumption of undercooked or raw pork, and a fish tapeworm, consumption of undercooked or raw fish. So a pinworm can cause a common infestation of the large intestine. The worms can be 2 to 12 millimeters long. They have a tapered, curved, cylindrical shape. Simple, uncomplicated infection does not spread beyond the intestine. The life cycle of a pinworm, a roundworm. So swallowed of the well, eggs emerge. Let me start here. Eggs emerge from the anus. Scratching contaminates the hands, and the person can then reinfect themselves by swallowing the fertilized eggs. Helminth classification, identification. So characteristics such as shape, size, the degree and development of organs, the presence of hooks, suckers, or other specialized structures, modes of reproductions, kind of host, microscopic identification. Sorry, someone
Mute your mic.
needs to mute their mic, please. Somehow I lost there. Parasitic worms, there's about 50 species of parasitic worms that can parasitize humans. They're distributed in all areas of the world. They may be geographically restricted. Higher instances are found in tropical areas. The yearly estimate of worldwide cases is in the billions. Conservative estimates of just 50 million infections in North America. Humans have evolved in the constant presence of these worms. Only recently have humans developed a helminth-free existence. Absence of helminth infections may contribute to autoimmunity and allergy. So oddly, an infection can bolster our immune system. So when we compare these major groups, protozoan, fungi, algae, helminths, and humans, protozoan are always single cell. They do not have a cell wall. Their cytoplasm can be divided. They can use flagella, cilia, or pseudopods, or none for movement. They can form cysts. Fungi can be single cell or multicellular. Their cell wall can be chitin or sometimes cellulose. They do not have, only the protozoans can have a divided cytoplasm. All of these groups are heterotrophic for the most part. Some algae and protozoan are autotrophic. Humans obviously can move by limbs, and the protozoan, fungi, and algae and helminths can all have flagella, though the fungi, algae, and helminths, the flagella is seen in the gametes. So we're going to talk about about the difference between nematodes, cestatodes, and trematodes. And I want to make a little note about that.
Isn't it the
Okay.
shape?
Pardon?
I was saying if it's not
Pardon?
the shape that differentiate them.
I'm sorry, I'm not quite getting your question.
I'm saying like, isn't it like the shape that differentiate them?
The
The shape.
She said the
shape.
shape.
The shape.
The shape of a person.
No, for the description between nematodes, cestodes, and trematodes.
Oh, the shapes. Yeah, we're going to talk about that in a minute. I'm sorry, I was making a little note there. In elements, the most developed organ system is the digestive tract. Did we talk about the difference between the nematodes, cestatodes, and trematodes? Are we missing a slide here? But you were right about one classification criteria: shape, size, degree and development of organisms. I don't remember a slide specifically with these differences. Okay, I'm going to, let's take this is actually the last slide. So I'm going to take a 5-minute break and we're going to come back in 5 minutes so I can take a quick look at this. So it is 8:36. Let's say we come back around 8:41. Also, did Felicia Adams show up, or who was the other person? Piaja
Yes.
Talley? Felicia Adams is here.
Yes.
I'm here too,
And
Professor Piaja Talley. Did you hear me, Professor?
yes.
Okay.
And there was one other person, last name Balde. Bold.
That's me. That's me.
Oh, okay, sorry. I was
It's-
listening to you and looking at something else.
Yeah.
All right.
Allow you, she knows, yeah.
Yeah, I have everybody now marked as present. Okay, so let's come back and let me double check the time. Let's come back in about 5 minutes. Roughly 8:48. I want to look up something. Okay, I just want to, this will be the last thing I want to go over. Maybe I missed it on the slide. But the nematodes, cestodes, and trematitodes are all parasites. The nematodes are the roundworms. Cestodes are tapeworms and have a ribbon-like structure. The trematodes are what are called the flukes, and they are internal. All these are parasites and are fairly widely distributed. A lot of them are found in the soil and in water. Okay, so before we stop, are there any questions? We have exam 1. It's going to be next week. It'll be available for 24 hours starting Tuesday, February the 15th. I'll put it in the morning and it'll be available going into Wednesday. 50 questions, true, false, and multiple choice mostly. Does anybody have any questions?
Will there be any short answers, or it's just the multiple choice and
I believe
the
it
other?
is all true, false, multiple choice. There's no written part to the exam.
Okay, thank you.
Can we have a study guide though?
I'll post by tomorrow, I'll post an announcement. Pretty sure there was one, but I'll double check.
Okay,
And
when is the
post it.
exam? What time? Like normal class? time?
Pardon?
Is the exam go like the same time as the class?
The exam time, I believe, is an hour, maybe might be a little bit longer.
Maybe at 7
Might
p.m.
be, I believe it's 70 minutes for the exam.
Is it at 7 p.m.?
You
It'll-
said they have 24 hours to do
24
it,
hours,
right?
baby.
Oh,
24 hours.
You'll have 24
okay.
hours to do the exam. I'll post it Tuesday morning until Wednesday morning.
Oh, okay. Thank you.
Okay. I'll double check on the study guide. I usually put a list of the slides that I think are most important from each lecture. Remember, the chemistry is not on exam 1 and it's not on the final. However, there is on the Connect, there's a quiz and a smart book assignment for the chemistry. And there will be a lecture next week.
Professor, during the lecture next week, are we going to review anything for the exam or no?
No, the exam's that day.
There is a study guide. It's posted under modules.
Yeah, isn't it?
Review guide?
Yeah, doesn't it in the first module? I'll double check and
The
send
module
out
that
an
says week
announcement.
1.
What does it say again?
It's under the module that says week 1, and that little arrow, when you press it, it shows everything under there, and it says review for exam 1.
Okay, thank you.
You're welcome.
Thanks for pointing that out.
You're welcome. I have a question though. For Connect, when I finish those the SB things, it's not showing up in my grades, but can you still see that I've completed it?
Can you send me an email and specifically tell me which one, and I can look it up and then email you back?
Okay, I'll do that.
Sometimes it will take like a while to update.
I see what you're saying. What happens when you take a Connect assignment, I have to periodically refresh the grades. In other words, In other words, like, let's say you took a test, a Connect test yesterday, and if yesterday evening I had refreshed the grade, you would see it. If you took it today, I haven't done that, you wouldn't see the grade until I do refresh it. So I have to constantly do that. It doesn't appear until I've done that. So I usually do it every few days. Does that make
Okay,
sense?
thank
So
you.
it just
Yes.
means, yeah, so you should see it after a few days.
Okay,
In
thank you.
fact, I'll, after this, after this, I will go into the system and refresh the grades.
Is there a specific due date we have to do all the work by? Because mine said all of it is due by May.
All the Connect assignments are due by the end of the semester. You have the entire semester to do all the Connect assignments. So does anybody else have a question? So I'm going
What are
to
the
end.
chapters? I have one more question. What exact chapters are we studying for the exam?
So for the exam, it is chapter 1, 3, 4, and 5. 1, 3, 4, and 5 is exam 1. I'll post
We have
that.
to do the exam during class time, or you're just gonna open it?
It's 24 hours. If you have some odd reason in your schedule that you have to take the exam during class time, I use taking the exam for attendance. So once you've taken the exam, I'll mark you as being present next week. So if for some reason you have to miss next week, because that's the only time you can take the exam, you'll still be marked present that day.
Wait, so we have to take the exam during class time?
No, you do not. I'm just
okay.
saying the exam is still available for 24 hours. That includes, it'll be available during the lecture next week. I'm just
okay.
saying if there was some reason that was the only time you could take it, go ahead and take it. You'll
okay.
still be marked present because I use taking the exam for the role.
Okay.
Does that make sense?
It makes sense. Thank you.
We'll have a lecture next week.
All right.
Okay, any more questions?
That's all for me, Professor.
Professor, will we be able to go back and review this class?
I'm working on trying to post the videos, and hopefully I'll be able to post some more videos before the test.
Okay,
But you do, the PowerPoints are there, and there's a review for the test in module 1.
okay.
Okay. Any more questions?
It's 50 questions in 70 minutes, right?
Yes, I'll double check, but I believe so.
Okie dokie.
All right. Well, good night, everybody. See you
Good
next week.
night. Good night, everyone. Good luck.
Good night, thank you.
Thank you.
Thank you, Professor.
Okay, thank you, and good night.